Welcome back. Another exciting day in the world of biology of cells and tissues, talking about the nervous system. This is part 2, so of course, if you haven't checked out part 1, where we talk about kind of more the big picture of the nervous system, go ahead and go back and watch that one. This is going to be the part where we get more into an in-depth dive of the cellular anatomy of the nervous tissue. Of course, like anything, leave a like on the video. If you're not subscribed, go ahead and do that right now, as it really helps out. And let's get into it. So talking about the nervous tissue histology. So there's going to be two principal cells types that we're going to be talking about. We're going to talk about the neurons themselves, these nerve cells, the cells that actually are transmitting those electrical signals. And then we're also going to talk about the neuroglia, or the glial cells. Either way you want to say it is fine. These are a lot of our supporting cells. They're the small cells that surround and wrap around the delicate neurons. So we're going to start by talking about the neurons, and then we'll get into all the glial cells. So neurons, these are the structural units of our nervous system. They're large, they're highly specialized cells, and that conduct the impulses, or they carry those messages. They carry electrical and chemical messages. Okay? So specific characteristics that we need to know is that nerve cells, once we have them, they last for forever, or hopefully they do. Other issues, depending on how we treat ourselves based on, injury and how healthy we are, the things that go on throughout day to day, determine exactly how healthy and how many of them exactly last. But they are meant to last a person's lifetime. They are also amitotic, which means that they do not divide and they do not replicate. Okay, so once we have the neurons that we have, that is what we have. We cannot replace them. We will learn in the future that nerve cells can grow and they can heal to an extent, but we can never truly get new ones. Okay? And that whole, what I just said about they can heal and they can grow to an extent, kind of lengthwise and size-wise, that is going to be a much in the future discussion, more in trimester 4, trimester 5, when we dive deeper into neuroanatomy and clinical neurology. Don't worry about that right now. The nerves have a very high metabolic rate. They require a lot of energy and oxygen. So the brain itself uses about 20% of the body's total energy that we take in, even though it only makes up about 2% of our body weight. So lots and lots and lots of energy. So starting to look at the individual components, we're going to start with the cell body. This has a couple of AKAs. That's either the cell body or the soma or the perikaryon. We saw this word in the last video on one of our pictures. Perikaryon, again, same thing. Soma, cell body, it's all saying the same thing. This is the biosynthetic center of the neuron. So this is what we determine as the center of the neuron, kind of like the main portion of the neuron. This is where all of our stuff is produced, where the proteins are made, the membranes are produced and maintained, where the chemical messengers are made as well. We also have what is called rough endoplasmic reticulum in there. We've seen rough ER before, but we also get some special names for it. We can also call it, in the nerve cells, chromatophilic substance or Nissl bodies. Remember in the previous part, in part 1, we talked about Nissl staining, because that was the guy's last name, and he discovered these Nissl bodies. So Nissl bodies, chromatophilic substance, rough endoplasmic reticulum, all saying the same thing within the context of the neuron. The nucleus as well as nucleolus are going to be spherical within the nerve cell, and we have all of the organelles and stuff that we have before. So here we can look in these rough endoplasmic reticulum, aka Nissl bodies. We have a nucleus, we have all the other components, we have the mitochondria, we have the smoothie art, we have the Golgi, all of that stuff. We have all of that within our cell body. Now, coming off of the cell body, we can see right here, coming off of the cell body is a structure called the axon. The axon is how we transfer the message to other parts of the body. Even though the cell body itself, I'm going to call it the soma. That makes things a little bit easier. So that even though the soma produces all of these chemicals and everything, it's going to then send the chemical signals and everything down the axon in order for it to travel to other places wherever it needs to connect and pass that message along. So the axon, this is a long, slender projection of a neuron. It transmits the electrical impulses, which we also call action potentials, away from the cell body. They have a constant diameter, and each neuron has one axon that starts at a cone-shaped area called the axon hillock. So right here, this cone shape right there is our axon hillock. This right here is our axon hillock. In some neurons, the axons are short or essentially absent. In others, it almost comprises the entire length of the cell. So some axons can be over 1 meter long. Think about some of the nerves and stuff that we have that run from the spine all the way down to the to the big toe or into the foot. It's a long distance to travel, so we can have the soma of that nerve can literally be inside of our spinal cord, and it travels all the way down to our feet. So some of these axons can be incredibly long. The long axons we call nerve fibers, and we saw the overall structure of those nerve fibers back in here. Each of those individual nerve fibers all coming together to form fascicles to then form a full nerve. And we were here. Axons can also have occasional branches called axon collaterals. These are extra little branches, just like this. It's an axon collateral. And axons branch profusely at their end, at their terminus, their end, up to as many as 10,000 terminal branches. Those distal ends are called axon terminals or terminal boutons. So these terminal branches here on the very end, these are our axon terminals, or the terminal boutons. You can have tons and tons and tons of those. Just a couple more anatomy type stuff. Vocab words. Axolemma. Anytime you see that emma right there, think about plasma membrane. We're dealing with some type of membrane. Axo is the axon, lemma being the membrane. So it's the specialized plasma membrane that surrounds the axon. Axoplasm, just like cytoplasm, the gel-like substance found within the axon. Those are probably going to pop up. Each of these terms can pop up just as easily on test questions. Just understanding, what their name is and where they're located. So within the axon generates nerve impulses that transmits them along the axolemma, or that cell membrane, all the way to the axon terminals. At the terminal, you have neurotransmitters, which are chemicals that can be released and go on and bind to another part of a different nerve, a different neuron, in order to either excite it or inhibit it, depending on what neurotransmitter is released. That gets a little bit fuzzy, kind of talking all about that. You're going to go into a lot more detail of all of this in Physiology 1 next trimester. So what I want you to know for right now is that these nerve impulses, or these action potentials, travel along the membrane, the axolemma. When it gets to the end, it gets to that axon terminal, it releases chemicals that then bind and pass along the message onto the next neuron. So here's this picture, another picture to see all this stuff. There's the axoplasm, the axolemma, the helic, the axon collateral, and as you can see, there's a lot more components to the axon as well. Some of this myelin sheath, which we're going to talk about in a couple different places, just because there's a lot of components to that. So the synapse. So we follow the axon all the way along. We get to those terminal branches, those axon terminals, terminal boutons, and we get something called a synapse. The synapse is the site of contact between neurons and other cells. It's unidirectional transmission of nerve pulse, so it always goes in one direction, and there can be electrical or chemical involved. When I talked about neurotransmitters, that is our chemical, which is the most common version, but you can also have electrical through these things called gap junctions. You'll see more of that. Most of what we'll talk about and what you'll see is going to be chemical, so focus on the chemical side of things. This is just an example of a synapse where we have right here. This is our our axon terminal. These are the neurotransmitters. They get released, and they have a receptor on the other side where it will continue down. And where it connects to is a structure called a dendrite. So the axon terminals connect to dendrites of another neuron. Correction, they can synapse with a couple of different things. If they're synapsing with another neuron, they synapse with a dendrite. So dendrites, they can have hundreds of these. They contain the same organelles as in the body. They're kind of the input region of the neuron. And they contain these other structures called dendritic spines. Think of the dendrites as the information collectors. They receive the information from another neuron or from another source. These are our dendrites over here by the soma. And on the dendrites, they're covered with these dendritic spines. And see, there's the axon, which comes and connects to the spines to synapse. So a signal will come down the axon. A chemical will be released from the axon terminal. The dendritic spine has some receptors on it. The chemical is going to bind to those receptors, and it's going to cause a chain reaction, which then the dendrite sends the message along to the neuron, down to the soma, and then that soma, if there's enough of a signal, enough of a I'm trying to figure out the correct word to say here. If there's enough of a graded potential, so enough of the chemical release, enough of the chemical bound, then it will trigger what's called an action potential, that message, that chemical, the impulse, and send it down the next axon as well. Now, myelin sheath, this guy right here, that surrounds the axon. The myelin sheath is composed of myelin, which I mentioned a little bit about that before. It's a whitish protein and lipid substance, lipid specifically. So remember, our white matter in the brain and spinal cord has lots of myelin, and its job is to protect and electrically insulate the axon. By insulating the axon, we actually get to increase the speed of nerve impulse transmission. Just another picture of the neuron structure. You have all this. We're going to cover a lot more about the myelin sheath, where it actually is, what are like the little nitty-gritty components of it, as well as how it's produced later on in this PowerPoint. Okay. Again, there's a lot of moving pieces and different components, so I just want to keep similar material together. So this is kind of finally getting into the classification of the neurons. We've seen some of these terms. We've seen multipolar, we've seen pseudo-unipolar, we've seen sensory, we've seen motor neurons in the first chunk of material in part 1. So now we're going to figure out what those terms really mean. So the structural components. The structural classification of neurons. We have 3 different types of structures with neurons, and they're determined by their number of the processes or things that stick off of them. The multipolar have 3 or more, so 1 axon and a whole bunch of dendrites. Bipolar has 2, 1 axon and 1 dendrite. Pseudo-unipolar, and these are T-like, and they have 2 axons, what we call 2 axons. They're also called unipolar as well. Here is our multipolar. These are the most common and major neuron type in the central nervous system. You can see how it has 1 axon and a bunch of dendrites. Bipolar, more like the cell body is in the middle, and it has this 1 branch off of dendrites. So this is considered 1 dendrite, and then 1 axon. Pseudo-unipolar, the cell body or the soma is kind of off to the side, and we count this as axon 1 and axon 2. Even though it's sort of more continuous, but we say that it has 2 axons. There are all of those put next to each other. Make sure that you know the straight definition of each of these, as well as other names. Let me bring this to the front. There we go. As well as just the general information, the information that's on each of these slides. That's as much as you need to know for each of these for now. Here's just another picture of the multipolar versus bipolar versus unipolar. Functionally, they're split up between sensory, motor, and then also interneurons, so neurons that connect to other neurons. I'm going to highlight a couple of things here that I know likes to get asked. So sensory, they transmit impulses from the sensory receptors towards the CNS. Almost all of them are pseudo-unipolar, so that's going to be a big help answering questions there. The motor, these are multipolar. And then our interneurons, the interneurons actually make up 99% of all of our neurons. There's a ton of them. Mostly just within the CNS. And this is what they connect neuron to neuron, and they build kind of that neuron network to allow all the different components of the brain, of the spinal cord, to communicate with each other and to know exactly what needs to happen when. Again, here we have our sensory neuron, goes in through the dorsal horn. Interneurons connect neuron to neuron, and then our motor neuron comes out the ventral horn and makes us move. Okay. Well, I think what I'm going to do, I'm going to split this one up into 3 parts, because we only have about 10 minutes left, and there's no way we're going to get through all of the glial cells. in the next 10 minutes. So I'm going to make this a part 2, and then we will continue on with part 3. Thanks again so much for watching. Have a good day and good luck studying.